So thank you for the opportunity to speak. I'm a physics professor, and when I teach my freshmen, the first thing I teach them is kinematics. Kinematics is a description of motion. So today I'm going to talk about measuring protein kinematics, and I'll be telling you in particular about time-resolved distance distributions after blue light excitation of a photoresponsive protein domain at room temperature in solution. So, proteins are fluctuating molecular machines. There's lots of motion even in thermal equilibrium, and many members of this community, Jack Fried and many others, have done beautiful work examining these motions. What are the functional motions of these machines? So that's the question I'm interested in. And so my working definition of protein kinematics is time-resolved inter-residue distance distributions after the proteins are actuated. The protein that I'm going to be focusing on is protein domain is ASLOVE2. And so I'll briefly introduce that. This is a light, oxygen, or voltage sensitive domain of phototropins in plants and algae. Phototropins control aspects of photosynthesis, and ASLOVE2 is from oats, is the protein domain from oats. So upon absorption of blue light, a kinase is activated and then can begin phosphorylating ATP. These are important optogenetic tools that can render many non-photoresponsive proteins responsive to light. So here are some molecular dynamics simulations. I've already showed you the molecular dynamics simulations in thermal equilibrium. These are simulations after activation. Now it's difficult to do MD simulations that accurately incorporate the activation of the flavin. And so we're substituting a pressure jump, which we believe may activate similar motions. So if you watch this molecular dynamics simulation, you'll see that this J alpha helix, the red J alpha helix, will gradually, over the course of this 200 nanosecond simulation, begin to undock from the protein, and that undocking of the J alpha helix is thought to be central to the action of this actuator. So there are various approaches to protein kinematics. One can activate a protein and then freeze quench it after a certain delay, and then follow that measurement by Deer measurements, or as solid-state NMR, or cryo-EM. Now, there also are ways that I'm interested in is to avoid the freeze quenching. And so methods here include serial X-ray crystallography, which has atomic resolution and picosecond time resolution. Limitation, of course, as we all know, is that you must crystallize. IR spectroscopy and UV circular dichroism have very good picosecond time resolution. They probe secondary structure, not tertiary structure. Forced resonance energy transfer has single molecule sensitivity. In some ways it's similar in spirit to the spin labeling. There's challenging data analysis, but people are working actively on that. And what I will talk to you about is what we call time-resolved gadolinium EPR spectroscopy, rapid scan, operated in rapid scan, or RS-TIGR. And this is a hard problem, so we will need all of these methods and more. So here's a brief history of this. I was inspired by this classic work from the Hubble group, where they showed that EPR could be used to measure structural changes during the photocycle at room temperature. In 2006, motivated by a colleague in the chemistry department, Song-Yi Han and I began a collaboration to what we called film proteins in action with high field EPR. Daniela Goldfarb introduced us to spin labels, gadolinium spin labels, in 2010. Then graduate students Stephen Edwards and Jessica Clayton showed that distance measurements can be made and up to almost 4 nanometers and even at room temperature. In 2019, Janet Lovett's group led the development of a beautiful spin label gadolinium as TPA-TCN that we're going to be using. And this is a W-band graph of the absorption of this. And you can see that it has a very narrow central transition, much narrower than nitroxide. And this central transition gets narrower as you increase magnetic fields. And since this is a very narrow transition, these gadoliniums are exquisitely sensitive to the presence of a nearby gadolinium. What I'll talk about today are the measurements that Brad Price and Shiny Métis have done, and Johanna Schubert is taking over this effort. So these are measurements conducted by Jessica Clayton to calibrate the method. These are measurements on gadolinium ruler molecules that were manufactured by the group of Adelheid Gott. And so she manufactured these molecules with gadolinium spaced by a very rigid linker with varying lengths from 1.2 nanometers up to 4.8 nanometers. These molecules were immobilized by either freezing or dissolving in trehalose. And here are the spectra. So you can see the green is the monomer spectrum, and the black is the data, and the orange is a Pate convolution simulation. And so these are very sensitive to distance, and they're distinguishable. We get distance sensitivity out to almost 4 nanometers for this spin label with even at room temperature. Our spectrometer is, here's a block diagram. So it's a 240 gigahertz, 8.6 Tesla spectrometer. This is the 240 gigahertz source, goes through a wire grid polarizer. It's the radiation that interacts with the sample is circularly polarized. Half of that gets reflected in so-called induction mode and then goes to subharmonic mixer and digitizer. Antonin Soika and Brad Price designed this beautiful, Antonin Soika designed this beautiful probe where he put some quasi-optics actually in the magnetic field. So the sample is here, and there's a roof mirror that lets us, rotating it, lets us cancel the the copolar signal. And our samples are in rectangular borosilicate capillaries, have a volume of about a microliter. So here is a rapid scan signal from 1 millimolar gadolinium TPA-TCN. Yeah, Brad Price definitely carefully read all of the Eaton's papers and Mark Zeitlin's paper. So we're getting signal to noise ratio of 60. So rapid scan, as many of you know, we have a magnetic field, a modulation field here with a 50 Gauss amplitude. And these lines are sufficiently narrow that that's enough to get the whole spectrum. Here we have a signal to noise ratio of 60 with 250 scans, which corresponds to 10 milliseconds. So the experiments that I'll tell you about are on ASLove2. It was labeled at these two sites. One is on the red J alpha helix, and the other is on the other terminal of the protein on an A alpha helix. So I'm going to show you a few snapshots of the movie. So this is the EPR spectrum just before the laser is turned on. Here the laser's turned on, and you can see that the spectrum sharpens. And then here is 50 seconds after, 88 seconds after, 126 seconds after. We can follow this spectrum as it changes during the course of this. Now I'm going to play this movie here. And so the screen will turn blue when while the laser is flashing. So that's right now. You see it goes up and then gradually relaxes, 1 spectrum per second. On the right here, I've represented all of the data in this matrix, and this is where the laser turns on. So this is and then you can see this change that then we can follow here. So Brad did singular value decomposition on this time series of spectra, and there were three principal components, and the first two of them are time dependent and show an exponential behavior. So here's the laser turning on, and these two components are decaying exponentially. So based on that SVD analysis, our hypothesis is that there are two time-dependent principal components based on that we have a dark state and a lit state, each with its own distance distribution. So this alpha is the lit state population, which we're going to assume has an exponential decay. And the dark state will have a population distribution, distance distribution with a mean R1 and weight 1 minus alpha, and the lit state will have a distance distribution with mean R2 and weight alpha. And we fixed the Gaussian widths based on deer data taken in Song Yihan's lab. So in order to extract distances and distance distributions, we need to relate the CW-EPR line shapes with the gadolinium distance in the presence of tumbling, because the proteins are tumbling, and tumbling tends to average out the dipolar coupling. So we use PAIC convolution, and so we start with, if you have a measurement of a monomer, or a singly labeled sample, and you convolve that with a PAIC pattern associated with the particular distance, then convolving those two should predict what the spectrum looks like for a dimer, and so this is working quite well. Now, in the presence of tumbling, the PAIC pattern is narrowed. So here, this PAIC pattern in green is replaced, depending on the rotational correlation time, this gets significantly narrowed. So we've chosen this rotational correlation time, which is appropriate for the size of the protein that we have. And here are the best fit time-resolved distance distributions for ASLUF2 in solution at room temperature. So here is before the laser is activated, and then the laser is activated, we get some of the long distance distributions, and that gradually relaxes. The best fit parameters that we get from this are that the dark state distance is 3.2 nanometers, lit state distance 4.5 nanometers. We have low confidence in this number because dipolar broadening is not very big for this distance. The maximum lit state population is 90%. So this flash of light turns 90% of the proteins into the lit state, which is something that you can't tell without doing a mechanical measurement like this. And this is a simulation of the dark state distribution based on the X-ray spectrum, and using the software KaiLife to account for the rotameric configurations of our spin label, and it's quite close to the value that we measure. The residuals are small, so this is the fit, and these are the residuals. They're less than 5% and time independent. And the refolding rate that we measure is activated and has 15.5 kilocal per mole activation energy. So our next targets are a thorough investigation of ASLUF2. We'd like to look at what order various structural transitions occur in, and does the J-alpha helix remain helical as it unfolds, or does it actually sort of start to disintegrate, or as you'll see at certain times in this movie. And we'd like to measure multiple pairs of sites to build up a movie of this protein. I'd like to also look at kinematics of an entire phototropin to elucidate the activation of the kinase. There are many improvements that are needed for this method. We know we can improve time resolution. We'd like to mitigate tumbling, and I'll encourage you to see the poster by Johanna. And the outlook, well, I hope that more people will join the fun. These measurements are fast, so far limited by the photocycle, and so we can easily vary conditions. Moderate resources will be required to build such a system. If you have access to an old NMR magnet, 7 Tesla or 9.5 Tesla, I think that should be sufficient, along with some electronics. That cost is non-trivial, but we'd be happy to help. I think that sub-millisecond time resolution is possible. The current resolution is 10 milliseconds. We have no cavity in our system. Limit to time resolution in principle based on T2 is tens of nanoseconds. That will be hard to get. But microsecond time resolution should definitely be possible. And I would love to hear thoughts from the audience about potentially improving data analysis. And I'd like to thank you for your attention and look forward to your questions.
We have some time for questions. Left.
Thank you for the nice talk. When you have your dark and your lit state, you see that when you shine light on it, you get the lit state. But you don't see a gradual movement of the distance from the dark state to the lit state. But it's more like that. It's not like that.
Okay,
so what does that tell you?
Well, so we've made the hypothesis. The hypothesis that we're testing is that there's essentially an instantaneous transition between the dark state and the lit state, and our data is consistent with that hypothesis. We are not yet sensitive to actually, say, the unfolding time. The unfolding, we should be able to measure that, but that's on a sub-millisecond time scale. So if we want to see the dark state expanding to the lit state, we'll need to go faster.
So I do think that when you, it's really nice talk, but as love 2 of flavin, isn't it?
Yes.
Yeah. Do you think it could interact with the gadolinium?
That's a very interesting question, whether the flavin could interact with the gadolinium. We don't see evidence of that, but yeah, the gadolinium is this big fluctuating magnet right next to this flavin, and yeah, so I think that's an interesting question. I don't know.
So you mentioned 90% conversion with a light.
Yeah.
Maybe naive question, but the original signal before you put the light on still seemed pretty strong. Do you see that? Can you see that from your measurements, that it's really 90% conversion?
Well, that's what comes out of the fit. So we do a global fit of all the data, and one of the fit parameters is the fraction in the lit state with the broader distribution. And
but wasn't the peak at lower? I don't know if you can go back to it.
You mean this one?
Yeah.
Yeah. So I think this corresponds to about.
I guess that one there, that one trace.
This one is broader. This one on the right is broader. Yeah.
Okay. Do we have any more questions? If not, let's thank Mark again. Our next speaker